Chapter 1, Section 8, Improving Contrast in Light Microscopy. One of the ways we can improve contrast, aid in our ability to see these small microbes, because you got to remember these small microbes, the majority of them aren't going to have any type of pigmentation in them. No chlorophylls, no colored dye, no colored pigments, none of that. So for all intents and purposes, they are nearly invisible. They're see-through. So we like to add in dyes. Dyes are going to be some form of organic compound that is going. So we're going to use these dyes as a means for adding color to the prokaryotic cell. Add color to it allows us to differentiate what is cell, what is not cell. Now these dyes, some of them are just basic, what we can call basic dyes. There's some form of a positively charged, pigmented, colored molecule that's going to bind to the overall negatively charged cell components, nucleic acids, certain polysaccharides, to the surface of the cells. Great examples of these: crystal violet, methylene blue, safranin. These guys just stain cells. Boom. All cells will be that one color. So when you go to stain cells, well, you can't stain them in a test tube as they're floating in liquid. You can't stain them on a petri dish. So what you do is you take some of the microbes, the bacteria, you smear them on a glass slide, you let it dry. Here's the problem though. Even though it looks like my hands are touching, they're not. My fingers are not touching. There is a layer of oils. There is a layer of molecular water, keeping them from actually touching at the molecular level. Well, turns out the same thing we find when we smear the bacteria on the glass slides. Pasteur and some of the other early microbiologists figured out that if you take those slides that you smeared some bacteria on, you let them air dry, and then if you were to heat fix, not burn, not boil, just heat up that slide enough that it's going to drive that molecular water out from between the bacterial cell and the glass slide. So now they're touching on the molecular level. Those bacteria are now fixed. They are stuck to the glass. So now we can come in with dyes, we can come in with water to rinse and all that good stuff, and the majority of the bacteria are gonna still stay there. So as you see here from this picture from the book, figure 122, you'll see that all of the bacterial cells will take up whatever this dye is, and they will now all be colored. Basic stains are great, okay? Lets you tell size, lets you tell overall shape, lets you tell overall arrangement. But that's it. Well, what if I want to know more? About a century ago, a guy by the name of, had the last name Graham, cannot think of his first name right now, figured out that if you mixed stepwise some of those simple stains, you can come up with what we now refer to as differential stains. The bacterial cells are still going to get stained, but any differences, say between two bacterial species that are present, will be highlighted using this staining process. Turns out there's one big difference between microbes. We kind of split them into two camps. Has a thick cell wall, has a thin cell wall. Now, yes, there are a few that don't fit into those two camps, but those are a few outliers. The majority either have a thick or they have a thin cell wall. Graham figured out how to use basic stains to tell the difference. We refer to this as the Graham stain. Graham stain, you take the bacteria or a mixture of bacteria, put them on a slide, let it air dry, you heat fix, all of that. And you come in with the first stain, crystal violet, which I just mentioned a couple of slides ago was a basic stain, and this one stains everybody purple. Thing is, crystal violet is easy to get into the cell, and thus it can get rinsed out of the cell almost as easy. So, what sets Graham's apart from everybody else's stain was the fact that he figured out that crystal violet and iodine react together, and they form crystals. They form big purple crystals, or big blue crystals, sorry. All the bacteria get stained blue, step one. Step two, all the bacteria now with the addition of iodine get big blue crystals inside their cells. The third step is to rinse away. Rinse away what, you say? Well, any excess crystal violet, any excess iodine, and membranes. Turns out alcohol rips up membranes. That lipid bilayer just gets shredded in the presence of alcohol. You know, cell wall gets damaged to a degree too. Graham figured out that bacteria with a thick cell wall, even though their membranes get damaged to the alcohol, that thick cell wall traps those crystal violet iodine crystals inside, traps them in the cell wall. So that big thick cell wall collapses and now becomes this meshwork that holds the crystals inside the cell. The gram-negative bacteria, well, their membranes go and their thin cell wall, even though it collapses, it's not a big, it's not a very good meshwork because it was a very thin, not a big thick cell wall. Those crystal violet iodine crystals just get rinsed away. They go clear. Final, come in with safranin, another one that we talked on a previous slide. That's a basic stain. Everybody gets stained red. You look here, you see this picture of a mixture of bacteria that have a thick cell wall, bacteria that have a thin cell wall. Thick cell wall we refer to as gram-positive bacteria. They will stain a bluish purple. Why bluish purple? Blue for the crystal violet, red for safranin, blue plus red makes purple. The gram-negatives that have only a thin cell wall, well, all of their crystal violet gets rinsed out, so all that's left for them is the safranin, thus they are a red or a reddish pink. Bluish purple, reddish pink. Gram-positive, gram-negative. The phase contrast microscopy is a way of changing how the light reacts around the cell. This is what you would see with a normal microscope. All that light, remember, coming off the light bulb is very chaotic. The photons are moving this way and everything coming in different directions. Phase contrast uses circular filters, so only light that's going through certain paths, this path or this path, but not this one, make it through the filter, thus go through the specimen, up through the microscope to your eyes. Light, just our normal type of light microscopy here, Bright field, look at all this stuff here. You know, you can barely make stuff out. Yes, I can see the overall shape, but seeing internal stuff, a bit difficult. Phase contrast, well, the light's hitting and it's coming around the surface of. It's going through, some of it goes through, and then it's coming around any internal structures that might be there. Ribosome, protein complexes. You know, if these are eukaryotes, I forget what these are. Oh, yeah, they're eukaryotes because right there's the nucleus. It's wrapping, the light's coming around the cell, and it's coming around and highlighting the organelles. So you can see organelles, you can get a really good look at what's on the surface of the cell. Dark field, everything but the cell itself is now dark. It highlights the surface, and it gives it this halo effect. So that dark field. Light that's only scattered, hits and gets scattered, is what's going to reach your eyes. Also gives better resolution because it's not so chaotic, that light. Only certain light makes it through. It's great for observing motility. If you want to see flagella, you want to see cilia on the surface of. So you're not seeing what's around the cell. You're not getting, you know, so much there because of stuff flotsam floating around the cells. You're not going to see so much internal stuff. That gets dampened down, but you're going to see what's on the surface. So if there's any cilia, flagella, you're going to see their movements. You're going to see them moving very well. And then sometimes, you know what? Light, just not going to cut it. That light coming from that LED right there is all wavelengths of light. But if I was to sit there and only use certain wavelengths, maybe a blue, in the blue range. And the specimen I want to look at, I had added certain compounds that under blue light, when bombarded with blue light, will fluoresce yellow, or will fluoresce red or green. And those compounds have an affinity for DNA, have an affinity for certain proteins, or have an affinity for certain type of lipids that can be found in only certain membranes. Yeah. Then I can start to highlight certain things over other. I can see internal structures, I can see external structures.